Yes, but now we will be looking into asexual reproduction methods. I mean, the categories in detail. First, we start with fission. What is fission? Meaning splitting or breaking. What here? A parental cell splits into daughter cells. Generally, people say a parental cell which splits or divides into two daughter cells directly. So that is binary fission. But which you can say it splits into daughters. How many? If they split into a single parental cell, which splits into two equal halves, what you call as binary fission, right? If a parental cell which divides unequal multiple parts, it produces many multiple parts which are unequal. What do you call them? Multiple fission. Example, plasmodium.
Sir, but then how will you tell that they are identical? Because asexual reproduction means they're identical, right? Genetically and morphologically.
Right.
So in multivision, when you're saying that unequal division is there, then how
Very
will
good.
they be identical?
Very good. Unequal in the sense which is not exactly getting divided into two equal halves, which randomly gets divides into multiple unequal parts. Whatever it comes, that will become again later on. Once the division done, it will become as it is, as usual, as a parental cell. We'll go for a better understanding. Take amoeba, which has one nucleus, one vacuole, right, mom? You are clear, no? This, okay, I'll make it more clear. This red one is nucleus, just imagine. Now the pink one is vacuole. Clear? This is nucleus, this is vacuole. Now, if a parental cell divides equally, right? What it's supposed to get, mom? This left half and right half. Am I clear?
Yes, sir.
Right. Now look at the left half, and this is the right half. Now, left half in the sense like this cell, no, this will forms. But the nucleus also, it got half only, but it will become here full portion. Here also, nucleus is another half only, but it will become a full portion. But now what about vacuole, which only this particular right half got vacuole? Then what about left half? It should also have, no? So during its lifetime, it will acquire, it will develop. Have you understand now? Understand what
Yes, sir.
it doesn't mean now? Is it genetically equal or unequal?
Genetically equal.
Equal, identical. Only the thing is, you need to understand the concept. Here, this nucleus will get divided into two equal cells. The nucleus will become two nucleuses, one off to the one cell, another off to the other cell. So this one off nucleus will become full nucleus, and the other off will become other full nucleus. Now, when a cell get divides, whatever the one half of the nucleus this cell gets, and the other half of the nucleus this another half cell gets, will become fully formed. But if you talk about vacuole, if you consider the vacuole is which only one cell receives, but the other cell does not. But during its lifetime, even the nucleus also form. I mean, the vacuole also form. But are we saying this is genetically identical and morphologically identical to that of the parental cell? You are clear, no? Perfect, right?
Yes, sir.
The same way, if you talk about plasmodium multiple fission, which divides into multiple unequal halves, right? Now, this how many halves more? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 cells will be formed and they are unequal, but they'll become equal. That is its asexual reproduction method. And now this each and everyone will become a parental cell, parental individual slowly. Now again, this will get multiple unequal halves, and this will be producing that many. How many unequal halves? That many cells. Again, this will become a parental cell. Say multiple unequal halves. This will become a parental cell. Multiple unequal halves, parental cell, like that. The same here, amoeba. Yes, again, this will be equally divides. Nucleus, nucleus, and vacuole later will be formed. Vacuole will be received here directly. This is what a fission. Fission is nothing but where a parental cell gets splits into two or more halves. If it is two equal halves, which you can say to be binary fission. Examples for binary fission now?
The amoeba, paramecium,
Very good.
the yeast. Can you say yeast? Yeah.
Amoeba, paramecium, yeast, euglena. But most of the times yeast will be considered as budding. But why it is in binary fission? It also splits, break down the parental body and able to produce two daughter cells. But it's an exceptional case, amoeba, paramecium, euglena, etc. But if you talk about multiple fission, example, plasmodium. Now, can you say the binary fission is of two types? Longitudinal,
Transversal.
transverse. Good. What do you mean longitudinal, Ma?
Say lengthwise.
Example,
Sir, we can say amoeba, the
amoeba.
paramecium.
Hmm. What is this?
Say euglena.
Euclid. Now this half will become this left side of become one cell, and right side of will become another cell. Right, Ma?
Yes, sir.
Perfect. Fine.
Yes, sir.
Good. What is transverse? Example. Come on.
No. Like, can we say
Hmm.
uh, I have no idea.
Shall we consider amoeba transverse curve?
No, but amoeba is generally, what do you call it? It is not
Hmm.
symmetrical.
Not symmetrical.
It is
Good.
complex. So
Okay.
it can happen longitudinally and transversally, no?
Longitudinally and transversally. But can you consider paramecium then?
The paramecium will divide longitudinally. No?
Paka?
Yes, sir.
Sure.
Because it
Sure.
has. Yes, sir.
Paramecium will divide even transversally also. The reason. Understood now?
So if it is like that, then all of the given examples should be able to,
How,
divide
how,
transversely.
look at how, if it, look at here, look at this plane, look at this plane, and look at this. But this is if you talk about almost, right? No?
So if they have
And amoeba,
symmetrical,
if you talk about this can longitudinally, transversely. There is no symmetry. But if you look at paramecium, which is a ciliate like paramecium, which can undergo for transverse binary fission. Yes, yes, no, something you're talking.
so if they have a plane of symmetry, like in that plane of symmetry is present, so they can divide in that plane. Like
R-
if we take longitudinally and they're symmetrical when they're divided longitudinally, so they can divide longitudinally.
Right,
Yes, sir.
right. The transverse binary fission which occurs in a paramecium, there is no doubt. The reason, which is plane of fission. It's not about plane of symmetry, which is plane of fission. The reason, which it has anterior end and posterior end. Got it, ma?
Yes, sir.
Right. Chalo. The next one is budding. What is budding, ma? Formation of a bud. Where it forms? Formation of bud over the surface of a parental body. Right, ma? Which you can say to be it's an outgrowth. Right, ma? Which you can say to be it's an outgrowth or projection of a surface. Can you hear me, ma?
Yes, sir.
Good. Now, tell me the types of budding. Exogenous
In
budding.
get yeast and
Huh?
in exogenous we'll get yeast and hydra, whereas in endogenous we'll get sponges, right?
Okay, good. Exogenous is yeast and hydra. So that whatever we see budding in yeast, budding in hydra. Whereas endogenous is sponges, they are gemmules formation or gemmicups, anything. Good. Let us begin with exogenous budding. What do you mean exogenous budding, no?
So buds that are present outside of the body of the parent.
Right. Birds that are formed outside of a parental body. For example, this is yeast cell. Now the bird has been formed. Now this bird will be able to grow and develop. Then this bird will be detached from the parental body, and the detached bird will be now able to grow into new individual, and on which the bird will be able to form again and again. But it's only a single bird method I told you. But over the surface of a parental body, on the parental body, bud will be formed. Over the bud, one more bud. Over the bud, one more bud. What you call this chain of buds. So
So,
how
so the
many?
oldest will be at the outermost part, and the innermost will be the new bird.
Hmm, good. Which one will be outermost here? This one or this one?
So the first, like the outer, yeah,
This
that
one?
one.
Yes, good. This
Yes,
is,
sir. So the new one.
which one is latest, recently formed? This one or one or two?
The one. One is
One.
the latest.
Right. The but whatever the bird which is being formed, that will be moving up. Good. And the later the newly bird being formed bird, which will be attached to the parental surface. It doesn't mean it's like a bubbles. One bubble, whatever which comes out first out, it will go up. The next bubble. The same way the birds will be. Good. Right. This is a budding in yeast cell. What is yeast mode? Unicellular eukaryote. Right, ma? This will be reproduced by method known as budding, and where the budding will be, the formation of a bud will be over the surface and outgrowth. So we call this method as exogenous budding. But if you go for budding in hydra, yes, ma.
So budding and hydra will be also
Also surface,
exogenous.
airface, surface of the body, which doesn't mean exogenous. That is what we listed clear. Now, how the parental hydra?
So here the buds will be like a new offspring, right? In hydra.
Right, boards will be like a new offspring, but it will be attached. How long it attached to the parent? As long it is considered as bud only. The same color should be for the bud, but to understand, this is the bud. But now, whenever the bud grows in the growing stages, now the slowly grows by formation of tentacles. This structures we call tentacles. Thus, you call it as anterior end, which is mouth. Here also the formation of tentacles slowly, then mouth, then this detaches from the parental body. The detached body will become now a new individual. Right now, this body will be growing, growing. Then this will become a complete parental hydra like this. And again, on which surface it is able to form the new bud. Clear, ma?
Yes, sir.
Any confusion?
No, sir.
Clear, no?
Yes, sir.
Good. And this is budding in hydra exogenous. But if you take sponges, how it will be? Yes, ma.
Sir, I didn't get you.
If you take endogenous body, sponges.
The spores will be like, sorry, I mean the, what do you call that? The bud will be formed inside the body of the parent and then will be released out.
Okay.
I'm right, yes.
Yes, you are correct. Inside the body means how?
So like inside a body in a sense, It is
Hmm,
when the, what do you call that? If we take the internal wall, it will break, and
very
then the
good.
spore will, like if we especially take in the case of sponges or sponges,
Sponges.
from the internal wall will break, and then from there it will go towards its mouth and it will come out, and then it will start dividing, I guess, and then it will become another sponge.
Good, good. So you can say the sponge is like this, right? And which has outer membrane and inner membrane. Am I correct? Like this. I'm not drawing. All of them like will be. And this is you call it as micropyle. Next, this is inner membrane, and this is outer membrane. Right now, what about this? All the lines, whatever we get. Like this. They are spicules. Clear. Now what happens inside them? I'll take the different color. Yes, ma'am. What are they?
So these are the gemmules, right?
Gemmules. Nothing but otherwise called.
Gemme cups.
No, this structure you call gemmacups. This inside things you call it as gemmules. Otherwise, you can say to be there archaeocytes. Otherwise, you can tell them as archaeocytes. Clear, ma? Now,
Yes,
what
sir.
happens when the conditions are favorable? This will burst, the micropyle open up, then the spores nothing but the birds or the gemmules or archaeocytes will be released out. Right, ma? In such a way, the budding which is endogenously, intracellularly in a gemmate cup, a cup-like structure. So this you could see in case of sponges. Clear? Any confusion as of now?
No, sir.